What I want to do is talk to you a little bit more about some of the details of how images end up being formed with the electron microscope. And again, I'm going to talk a bit about sort of the more historical things. So the images I'm going to show you may still not be the most spectacular, although they'll be, I think, pretty interesting. It's a sort of a joke, right? To look at small objects with the electron microscope. I mean, since the thing is used always for significant enlargement, of course we're looking at small objects. But what I meant in that was the idea that you could look at large objects and make them small by cutting them up or taking smaller slices or doing something like that. We're going to talk about that part of it next time. But there's a whole world of really small objects that were visible only when the electron microscope became available. So I'm thinking specifically about things like viruses, or for that matter, proteins, small objects that are below the limit of resolution in themselves. There are a couple of ways of looking at these things, but I thought first we really needed to get at least a somewhat better idea of what the samples actually look like and what it's like to put something into the microscope. So this picture here actually includes one electron microscope grid, which is this structure right here, attached to a forceps. Okay, so this thing is about 3 millimeters in diameter, maybe even less. It's pretty small. And the grid itself is circular in most cases. And it has in it a whole bunch of cross bridges. So it itself has, which is why it's called a grid. And these things can be 200 or 300 apart. That is, you'll have 300 of them in an inch, if that's what the measurements would be. So they're very small intervals. And generally, your sample ends up in some way or other spread over the bars of this grid. Now, if your sample is viruses, or if your sample is protein molecules, and you put them on a mesh like this, they'll just fall right through. You won't have any support for them. So what becomes necessary is to take this grid. Now I'm going to give you a side view of it, and you'll have to imagine the grid bars. I mean, I can try and draw them, but it's kind of, okay? This would be a side view, and now you can see these bars and the spaces between them. What is necessary is to create a very thin film that sits on top of the grid. And it's that thin film to which you apply the material. So that film is generally made out of one of two things. The film can be made out of a plastic, Generally, there's a brand name called Formvar, which is the most common of those. Or you can actually make a film of carbon, and we'll talk about that in a second when we talk about shadow casting. And the idea, what's essentially impossible to see in this picture, is that there's a very thin film floating on the surface here. And it's so thin you can barely see it. And the idea is that you take a grid like this, and you put it on this surface. There are actually two ways of doing it. You could put the grid on top of this film and then somehow flip the whole thing over when you pull it out. Or you could try to slip the grid underneath the film and then pick it up through the film. In either case, you end up with a film on the grid in most cases. So once you have a sample that is sitting on the grid, now you can take, for most of the objects I'm talking about, you can take a small drop. If this is a grid, and I'll give you a couple of steps of the grid. Okay, what you then do is you put a drop of the liquid that you have your sample in on this grid. So we're talking about a microliter or two of volume. You either let it air dry or you remove most of it and leave a very thin film on top. Okay. And then you have several choices. One choice is to apply some sort of staining material. That would be, in most cases, this idea of negative stain. The other would be to let it dry completely and carry out this process of shadowing. Shadowing is a little different from what you do as a pre-med student. In this case, the shadowing means applying a coating of some material to the sample that's on the grid. So now, if I go back and enlarge this grid a little more, and say that I now have some samples on it, and I'll draw my sample as little balls for the moment. What I will do, if I'm going to do a shadowing experiment, is, and there's a wonderful diagram here, okay, is put the sample in a vacuum system, into a vacuum, it's generally called a vacuum evaporator. And your samples, your grids, are sitting here. So you have a series of grids over here, each one of which has your sample on it. It could be a lot cleaner, but that's sort of the way it is. And at some distance away, you have a pair of electrodes in the vacuum system. You have a pair of electrodes, and in the one that's drawn here, it's actually drawn as if it has a basket in it. So what you'll see here is a wire and then a kind of a coil. And then the wire continues. So this coil is referred to as a basket. And what you do is you put into this basket one of these metals, little bits of gold filings, platinum, palladium. We'll talk about carbon and platinum in a minute. And you can put them into this basket and then run pretty high current across this thing. This entire thing becomes extremely hot and vaporizes the metal that you've put in there. In most cases, that metal is evaporated and ends up coating the sample. So if I go now back to my grid, here's my grid, and here are these samples that sit on it. Now, what happens is, after this process is done, I get a buildup of material on the side of the sample that was towards the electrodes, so from which the vapor was coming. And it might build up a little bit on top. And then if I were to look at this from above, so let's now say we're going to be looking down on this grid that has these circular objects on it, spherical objects actually, okay? The coating will end up here and here. But of course, it'll also be all over the grid, all over your film, except for where your object blocked it. So there'll be a region like this that doesn't have any of this material in it. So it will look as if you have an object that's being illuminated with a, say, a flashlight, and it casts a shadow, and that's where you get that impression, okay? So let's see if I can show you both some earlier and maybe some later shadowed images. These are from, both of these are from pretty early research papers. This one by Robley, Williams, and Dempster is actually a teaching tool. And what they've got here, there are two samples, very low magnification. This is erythrocytes. Here. And this one, believe it or not, is paramecium. Okay. And what has been done here has been ridiculously little. And that may be part of the problem with why they show up the way they do. Okay, so let's start with the erythrocytes because that's a little more familiar. And you can see each one of these is an erythrocyte. And you can see this sort of double concave shape. Well, you can see one side of the double concave shape. And there's a couple of very interesting aspects to this. Notice, first of all, that the way this image is presented, the shadow, that is to say, the area that didn't get coated, is black, okay? And the background, which does have some coating, has some grayness to it. And the area that was close to the electrode, so I'm assuming the electrode was actually up here somewhere, and the direction of the flow of material is like that, okay? So it builds up on one side of these erythrocytes, just as I drew it. And then on the other side, there's much, much less. And then there's the area where there's none at all. And so this does give you a very interesting three-dimensional sense of the material. What they've done in this paper is to confuse you a little bit. They've taken this same image and they've basically reversed it, made a mirror image, which is sort of odd. But then they've also reversed the contrast. So now you see white shadows with black samples, okay? What's interesting here is that then you go and look at this picture of the paramecium. Notice there are no magnification scales in here. So, you have to know this is going to be, what's the size of a paramecium, right? It's certainly 10, maybe 20 microns. And there's a substructure that is visible here, which is probably the roots and the structures of all those cilia that are bound into the paramecium, but normally you don't see it. And once again, you have this interesting shadow effect, in which case here you'd assume that the direction of shadow is like this, the direction of evaporation, okay? Whereas in this image, it's going the other way. Well, Okay, so that's one way of looking at it. But in fact, it was being used. This is 1948, before a lot of the techniques were available. Keith Porter, whose name you'll hear a number of times during the next few lectures, Keith Porter started to look at cells that might be making virus. He was particularly interested in the idea that certain viruses, especially those that caused cancer, that there were viruses that would cause cancer, and that those viruses could be visualized. Okay, so in this pair of pictures, and not a true pair, in this set of pictures, these are pictures of actually cells from a mammary cancer in mice. Okay, there had been a lot of circumstantial evidence that there was what was called a filterable agent initially, which meant, for many people, meant a non-even bacterial sample, therefore something like a virus, that could be transmitted in the milk of mice, certain strains of mice, that would give the daughters of that mother, would give them a propensity towards developing breast cancer. And so this thing was originally called the Bittner agent, eventually became known as the mouse mammary tumor virus. At any rate, what Porter and Thompson were trying to do was to see if they could visualize these viruses. And they showed, for instance, if you take some of the breast cancer tissue, you spread it out. You spread it out on a EM grid. And in this case, I think they stained it with osmium tetroxide. We'll talk more about fixatives later on. But osmium was a well-known fixative at that point. And you dried it down, you could see these kinds of particles. And they thought, well, gee, maybe that is viral material. And then they tried shadowing, and they saw a set, for instance, over here, of particles that were sort of standing out on the cells that they were looking at. And they were showing them, as you can see here, most dramatically by using the shadowing technique. But then it got to be much more sophisticated. Okay, this is some work from Henry Slater and Susan Lowy from about 1967. And some of you may remember in the cell structure course, I talked about Hugh Huxley's experiments with isolation of myosin and characterization of myosin in terms of the muscle structure. Slater took this quite a bit further and analyzed the structure of myosin. This is where you can actually see the two heads, for instance, over here. The two heads of the myosin molecule in the standard form of myosin. But there's another trick that was played here. The images in the first column were all done with shadowing the way I described them. That is to say, they were shadowed in such a way that the shadow came from one direction. Now, these are individual pictures, and they have carefully lined them up so they look about the same. But the idea is that the direction of the shadow in all of these three is roughly this direction. Okay? And the question that they ask in this paper is if you do a one-directional shadow like this, can you make out the substructure of the myosin head? And their answer is, well, you can really stretch it. Maybe you could imagine once in a while you'll see something like this where there might be two pieces. But by and large, the shadow tends to obscure the secondary detail. And so you can play the game a little bit more by saying, let me change the angle at which I do the shadow. So remember, if this is my structure, and I've got a grid over here and a filament over here, and I'm shadowing it this direction. If my object is fairly big, then a shadow is okay. If the object is really tiny, I may not see much of the shadow. And the way to get the shadow is to move the filament way down so that you're shadowing at an angle of, say, 6 degrees rather than 20 degrees or more. Okay? So that what this is like is if you're looking for something that you've dropped on the floor and you can't find it with normal room lighting, if you take your phone light and bring it way down to the floor, you'll emphasize the shadows, of course, of all the dust, but maybe of the object that you care about. So you can do a low angle shadow and see a little bit more that way. The other thing that developed was the idea of saying, well, supposing I slowly rotate the sample as I apply the shadow. So the idea here would be I put this on a little table. The table sits on a motor in some way or other, and the motor makes this thing rotate. And while it rotates, remember I've got grids on here with my samples. And while it rotates, I have my electrode over here releasing shadowing material at a low angle. And depending on how long it takes to evaporate the material and how quickly the motor turns and all the rest, I can get an image like this in which you actually sort of don't make out much in the way of shadow. But what you do see is this area that's been enhanced by, in a sense, being shielded from the shadow as it goes by. So you might imagine what's going on here. I go back to a little ball on the surface. And I start applying shadowing material to one side from a low angle. Remember, we're talking about a low angle. And then I rotate, and this object now starts to build up shadow on this side. You'll see that what goes on is that the middle has no coating. And so the middle of that sample over here, whichever one I'm looking at, okay, shows up as discrete particles. And now I can pretty easily see that there really are multiple, in fact, 2 heads on the myosin molecule. So this idea of rotary shadowing became a really important contribution. Well, this was actually started by a man named Kleinschmidt. And Kleinschmidt published this using DNA as a sample. And what he was able to do is, as you can see in this picture, it's been a little hard for me to show you these images because when you download the papers now, the copies in the digitized versions aren't very good. I've modified this one a bit so that we can actually see here this sort of line that represents DNA. And you can actually see that it curls around. Okay. Well, Kleinschmidt did one extra bit to this. He didn't just drop the DNA down on the surface. What he did was he added to the DNA some protein. Interestingly enough, he chose a very small protein, which was cytochrome C. And the cytochrome C basically lines up along the filaments of the DNA and causes them to spread out. That's what Kleinschmidt did. What he showed was that he could actually take a population of DNA fragments of some sort and show that he could trace them and figure out their lengths and see what kinds of relationships he was getting among the lengths. And what he actually claimed was that he was getting things that fit a kind of a sequence that went almost like a Fibonacci. 1, 2, 4, 8. Okay? Relationships from one to the next. And what he was doing was actually taking DNA strands. This is before we all fully appreciated the DNA structure. What he was doing is taking these DNA strands, which were pretty much chromatin, that still had the histones in there, which are in the nucleosomes, and looking at pieces of DNA that are broken off at different numbers of nucleosomes. Kind of an amazing bit of work. Well, that kind of thing can now be expressed, has been expressed even more interestingly in a case like this image I have on the right, where again, single-stranded DNA is incubated with a specific protein called PC4 protein. Okay? And the PC4 protein has a very interesting effect on the DNA structure. This is single-stranded DNA. When it's in the presence of the PC4 protein, you get this thing forming loops. But without the PC4 protein, it's just stretched out as linear molecules. So this PC4 protein has a role in the configuration of the DNA. So that's a sense, at least, of some of the power of shadow casting. And I'll show you more of it and more of its power when we get to talk about freeze fracture of all things, other techniques. Okay, but first I want to talk about today, at least, I want to talk about negative stain. And negative stain has an extraordinary advantage. The basic idea of negative stain is that you take a solution, you apply your sample to the grid on a film, just as I said before, and then you remove the liquid and then add of one of a number of compounds. The one that's most common is phospho. Sodium is a relatively neutral sodium phosphotungstate. Now it turns out, because of the tungsten that's in that molecule, okay, tungsten is a heavy enough metal that it actually can block a certain amount of electron flow. So that if there's tungstate present in the sample, you will see it as dark material. So it acts like a stain, except that it doesn't stain the material that you care about. Other solutions that will do something similar are uranium, uranyl, acetate, and instead of acetate, there's a molybdate. There's several of these solutions that can be used. Now, what Hall saw, this is an interesting side issue. Hall published a major paper in 1955, about the densitometric analyses of viruses. This was a virus called bushy stunt virus. Doesn't matter what it was. And as part of the paper, he said, he hadn't been using this neutral sodium phosphotungstate, but at one point he tried some. For most of the paper, he was looking at stained virus, but then he published these two images at the very end of the paper and said, under certain circumstances, this sodium phosphotungstate, when it's neutral, people had always thought you had to use it as an acid. But if you use it as a neutral, it doesn't get into the sample. And so when you have a tightly packed array of the viruses, you can see this kind of area here where the space between the viruses has got dark material. And so it enhances the general appearance of the virus. But what was more dramatic was that what he said was, well, I didn't wash it completely. Okay? When you don't wash it completely and the virus and the stain dries down around the sample, then you get this very interesting effect. So let me sort of finish my last bit here. After you add the drop, remove the drop, and allow to dry, okay? So that you still have a little bit of this stain left. And then you get this kind of interesting effect. So now let me see if I can draw on my extraordinary artistic skill. Here's the surface on top of the grid. Okay. This will be a virus particle of some sort. And this will now be the no, I don't like that color. This will be the phosphotungstate. So it will have wet the sample, presumably, and it dries down, leaving something like this behind. And what you see here then is this equivalent, a white sample with dark edges. Now, this was done in 1955, and the resolution of his microscopes wasn't particularly impressive. But as this procedure was applied to many different samples, let's see what we can get out of it. Brenner and Horn. This is Sidney Brenner, who was one of the founders of the field of molecular biology. In this case, they were looking at tobacco mosaic virus. At the time, that tobacco mosaic virus was a real concern because tobacco, of course, was a major crop. This is in the 50s and 60s, and anything that would damage the growth of tobacco plants was a serious problem. Anyway, there was a lot of support for research in this virus, this tobacco mosaic virus, and it turned out to have some very interesting properties. One of them is, as you can see here, its shape was as a rod, like this. And if you have the right imagination, you might say, gee, there's something in the middle of that rod, that it's actually cylindrical, although it's kind of murky. Well, the reason it's murky is that it happens to be filled with DNA. And so what they showed then was that if you now take this material and remove the DNA, Now you can really see the hollow space. And sometimes you'll get little fragments like this that you can actually almost sort of look your way down the structure. And if you look closely at a region like this, that is a little side view, you might imagine that what you're seeing, something maybe that looks like this. With the idea that this would actually go all the way around and form some sort of cylindrical structure based on these subunits. Okay? A little bit more sophistication, and you can get something that really starts to look like this. The picture on the bottom is a model. The picture on the top is actually photographically processed. And what they've done is they've taken a segment of this picture which they said, yeah, it's a little blurry, so I can't be too good about it. But if I take this segment and then take a segment of it and then move it over and place it right on top of the next segment over and photographically merge those things together. That was the old days where you merged them photographically. Now you can do it digitally. What happens is you preserve the data component, the things that repeat, the things that are built into it, and you get rid of the noise so that you actually end up cleaning it up. This image was actually prepared that way so that it's much cleaner, say, than these images. There's a lot more detail in them. So even at this time, people were manipulating these images. There was a man by the name of Marton, called now the Markham technique. Which was a photographic reinforcement. Okay. One sort of cute alternative to doing this sort of thing is if you had a sample that you were looking at, like one of these circular ones, as long as it was a pretty clean circle, and you suspected that it had a periodicity to it, you could mount it on a turntable again. Here's a turntable. Put your sample dead center, or put a photograph dead center on that turntable. And now start spinning it. And you may be aware, when you watch a car go by that has good bright hubs, wheel hubs, and suddenly you see the wheel hubs line up because it's a matter of how fast they're going compared to the periodicity of the light you're looking at it with. You could actually see the repeat pattern in samples this way, photographically. I'm saying this because I used to do it. It's a very interesting exercise. And at that point required no computer processing at all. All right. One of the kind of profound uses of negative stain was to look at some of the more involved viruses. A lot of the animal viruses are, in a certain sense, boring. Even coronavirus. I mean, it's basically a spiky membrane. But the structure of bacteriophage This is the bacteriophage, probably T4, that was the basis for, again, an enormous amount of molecular biology. Is shown here both in a shadowed version and over here in negative stain. Okay. And you can see the extraordinary amount of detail that is revealed by this. A bit of structure of the head, even in these sort of not particularly great electron micrographs, you can still get the sense of the head. You can see how the stain built up along this structure. So this is the stain in the format that I suggested before, forming a kind of a shell over it. So, okay, if this is the head, the stain is kind of grouping up this way. Okay. On the other hand, the stain is easily able to enter portions of the structure of this sheath, which is supposed to be the thing that actually squirts the DNA into the bacterium. Okay. And to show you quite a bit of detail of the hollow core and even these attachment fibers. So you could get quite a bit of detail out. And they showed that if you can fractionate this virus into its subparts. So here's the head, here's the sheath, okay? Here's this entire piece with the tail attached to it, and here are the attachment points. You could really check your isolation by looking at the morphology of what you got. And here's just another couple of pictures to show you as things get improved both in technique and in microscopy, we start to really see the kinds of detail that become available through this process of negative staining. And I had to include this picture, which is bacteriophage, all bound to a bacterium. So it's the sort of thing that is worth 1,000 words. It's a very dramatic way of imagining what a bacteriophage attack on bacteria might be. Okay. And then here's an example of what happens when you start refining this to look at protein structure. Okay. And this is not the best by a long shot, and you will see by the end of the course, we'll talk about cryomicroscopy, which builds on images of this, roughly this sort, which are isolated proteins, and you can see different arrays of the proteins. So this is one, presumably the same protein molecule, either sitting on its tail or lying flat. Okay, so this thing is essentially a little cylinder. Okay. And a cylinder with a hole in the middle and several different kinds of protein or different structures around the side, like that. And so depending on which way you orient it, you can see it in different perspectives and then try to generate a three-dimensional model of it. That's what's now being done in cryomicroscopy, bioelectron microscopy. This is another example of how you might be able to take a series of not quite so well-defined molecules, but by superimposing them, create a kind of a set of views that makes sense of that protein. Okay? And then I want to show you one more, almost as a warning. And I'm afraid this is a personal story. When I started doing work on, as it turns out, on the mouse mammary tumor virus, we looked at isolated virus by negative stain techniques primarily. And what we found as a characteristic structure for the isolated virus was something that looks like what you see in figure 1a. That is to say, it was something with a head and a tail, and a series of spikes all the way around this virus. There was a variety. This was the one that caused mammary cancers in mice. There was a parallel one that caused leukemia. And the leukemia virus, which is in 1b, looked rather similar, not exactly the same. Okay? Had a different membrane structure. And no spikes. Okay? Well, at one point I started wondering about the origin of this shape. What gives you the shape? And I realized that, in fact, what was happening was the stain, as it dried down, was creating a hypertonic environment and shriveling up these virus particles. And so there's a separate question. If they shrivel up, why don't they shrivel up into a prune? Why do they shrivel up into this kind of structure? But that seems to be what you got. What I did was I changed the procedure so that we fixed the virus with formaldehyde or glutaraldehyde, I think is what we used. And glutaraldehyde not only preserved the proteins better, which you can sort of make out, but also broke down that osmotic challenge. That is, they made the membrane permeable. And so what you ended up with was spherical particles rather than these peculiar tadpole-like things. And it turns out this is the structure that one sees in section tissues. It's the sort of structure that now, if I go back to this picture, that corresponds comfortably now with the sort of thing that Porter and Thompson saw, 30 years earlier. It's an interesting kind of path to have gone through. Okay, so I think I can stop here. Next time, I will be talking about looking at sectioned material and how we eventually can start to look through things.